if you have a specific question, we can do that. We can also just kind of work our way through from the oldest to the newest, if you have a preference. I have no agenda. I've seen the answers.
I think we should go oldest to newest.
Oldest to newest? Okay, we can work through that. And as we go along, if something doesn't make sense, just ask. In the interest of time, I'll probably move fairly swiftly. So if you want to, I mean, you're obviously always welcome to audio record if you want. But I will move a little swift so that we can hit everything. Okay, so if we go way back to the beginning, that would be chapter 2, not 3. Oh, look how far we've come. Oh boy. The days of the chicken till now. I better hydrate. No, turned out it was a one-time thing. I think it was food poisoning. Yeah, well, it was better than being sick for days, so I'm back on my game. A couple pounds lighter. Oh well. All right, so starting at the beginning, the location of subatomic particles, charges of particles, atomic mass, atomic number. So some of the first central and very basic chemical things. Make sure you're really comfortable with that. So of course, the subatomic particles, actually let's look at helium so we can actually see all three. Remember, the subatomic particles consist of protons, neutrons, and electrons. And you'll have to be comfortable with their charges. I think you probably are at this point. Neutrons do not weigh into the charge category. They simply add mass to the atom. But protons are positive, electrons are negative. They're located in such a way that the particles that have the most mass make up the nucleus, so the neutrons and protons. The particles with the least mass, the electrons, exist at predictable places around the nucleus, right? Of course, all of this in the context of real nature is not true, right? This is just a model to help us understand and discuss things, but in reality, it's much more complicated how the electrons sort of orbit that nucleus. Okay, so we know that in terms of atomic mass and atomic number, we can look at our chart here, courtesy of Dimitri, and the atomic number is always going to be the number of protons. So if we look at the center of our little drawings here, 6 plus 7 plus 8 plus 9 plus, that's a reference to the protons. It is also the atomic number, 6, 7, 8, 9, 10, etc. There's nothing about the periodic table that you should be memorizing for the purpose of this exam. Hopefully not for any class, right? It's a reference that we always should have. But make sure you understand how to read it. So if I ask you questions about this, I'll provide this context for you, and then I'll make sure that you can interpret it. So atomic number is the number of protons. If we change the number of protons, we change the identity of that atom, right? On the other hand, we can change the number of electrons, which will change our charge. So if we have more electrons than protons, we're going to have an overall negative charge. If we have more protons than electrons, we'll have an overall positive charge. If we're neutral, our electrons will be the same as our protons. Okay? And we can have different versions. We can have isotopes. So one of the questions here is be able to identify atomic mass number. There's that. I skipped the orbitals. Let me go back to that. So how are the electrons organized around the atom? We kept this at a very basic level, okay? It gets crazy complicated when you get past the first few energy levels, but in our case, the things that we want to know to understand most of the biology that we'll talk about this semester is that in the first electron shell, the maximum number of electrons we can have is 2, right? They exist in an s orbital, and the only cases where we really have to think about atoms that simple are when we talk about hydrogen and helium. Because after that, we add a third electron shell, and then things get slightly more complicated. But that first shell, just 2 electrons. Now, if we go to the second shell, we can have up to 8 electrons, right? And if it has 8 electrons, that outer shell and that atom itself will be very happy. It will be in a nice state, and so we'll see why that affects the formation of bonds here in a second. But 2 in the first, 8 in the second. The second orbital has, in addition to the s the second electron shell in addition to the s orbital also has some p orbitals. And then again, things get more complicated after that. I'm not going to go into it in this class. Not qualified to teach orbital chemistry. I haven't looked at that stuff in 20 years. But this is important for us to know. We want to know the s and p's, and we want to know that there are 2 in the first, and in the next few electron shells, 8. And trying to have a full electron shell is a goal that atoms are going to pursue when they form chemical bonds. Okay, now let's get back to our isotopes. So isotopes, where do I have? Here's an example of an isotope. Isotopes are where we have different numbers in the atomic mass than we see normally on our periodic table. So for example, phosphorus, number 15, usually has an atomic mass of 30.97, so 31, but phosphorus 32, which is a commonly used radioisotope in experimental biology, has 1 more neutron than normal, and so it's heavier. And so what will happen then is the phosphorus will decay because it is if it's a radioisotope, that means it's an unstable version of this atom, and so it will decay, shed radioactive particles, and become something else. So phosphorus becomes sulfur. And if we look at sulfur, its atomic weight is always, well, not always, averages at 32. So that's where it should be, not where phosphorus should be. So you have these unstable versions. If their atomic number changes, their identity changes. But if we change the atomic mass, it's still a phosphorus. It's just an uncommon radioactive phosphorus. Okay. Then what are the relative abundances of carbon, hydrogen, and oxygen? See table 2.2. This is a table that I'd like to ask questions about because especially this first section, the relative makeup of our bodies and our cells and the contributions that oxygen makes, and for example, that hydrogen makes. Let's look at that one. 63% of our atoms are hydrogen, but only 9% of our mass, right? And why is that? What is it about hydrogen that makes up so much of us, but contributes so little to our mass? It's light. It's a little baby. It's just a tiny little guy. And so the percentage of atoms versus the mass contribution are a little bit different, but these are the main hitters: oxygen, carbon, hydrogen, and nitrogen, right? That makes up the basis for most of our biomolecules. All right, let's see. What is a mole and how many grams are in a mole of a particular element? How many particles per mole? Oh, the joy. There's little Avogadro giving you the side eye. So a mole is made up of a specific number of individual particles. Now this could be individual atoms. So if we said a mole of carbon, those particles are carbon atoms. If we said a mole of carbon dioxide, those particles are individual carbon dioxide compounds, right? So 6.022 times 10 to the 23rd particles of carbon will give you 1 mole. And 1 mole of carbon will have the mass of 12 grams, right? So there's no calculation unless we have multiple carbons in the compound or multiple oxygens or whatever. If it's just a mole of an element, we look at the atomic mass and we put a G next to it. Now we did a calculation which I calculated wrong mathematically. Let's do a simple one. That'd be easier to not make a mathematical mistake. Let's ask if we want a mole of carbon dioxide. A mole of carbon dioxide will have how many grams of carbon? 12. And how many grams are in each oxygen? I guess we can't see it there. Let's look up our chart. I would give you this so you don't have to crane your head to look up that little number there, but how many are there?
16.
16, but we have 2, so we'll multiply that by 2, and we'll end up with 32 grams of carbon dioxide. So a mole of carbon dioxide would be 32 plus 12 would be 44 grams. Okay, so that's what we're doing with moles. And it would have 6.022 times 10 to the 23rd individual CO2 units within it. Nothing more than that. You can get really complex with mole calculations, but we don't need to do that. That's not the point of this class. That's the point of your 111 and 112 classes. Okay, what are the valence electrons and how do they influence bonding between atoms? What are the characteristics of covalent hydrogen and ionic bonds. All right, so let's switch our thoughts to why atoms would form bonds. Atoms are trying to form bonds to become stable and happy. And to be happy, they would like a full outer shell. So depending on what type of atom we're talking about, this will involve different things. If it is a fluorine and a hydrogen, which are trying to seek happiness, they can each share an electron with the other, and this gives fluorine, which had 1, 2, 3, 4, 5, 6, 7 valence electrons, 8. And hydrogen, which has 1, now has 2. So you can share an electron, and both atoms benefit from the contribution of that subatomic particle. So in this case, you have a sharing of a sharing of electrons, which means it's a covalent bond. If we look at some other atoms and how many electrons they might need, so oxygen has 1, 2, 3, 4, 5, 6 valence electrons, so it's 2 away from getting what it needs. Nitrogen has 1, 2, 3, 4, 5, so it needs 3. Carbon, our magic atom, has 4 in its outer shell, which means it needs another 4, which makes it the basis of organic life because it can form 4 bonds, which it provides an opportunity for so many different geometrical structures and so many different molecules. Okay, then if we think about, again, the types of bonds, the one we already saw was covalent. Here's another example of a covalent bond between 2 oxygen atoms. So you can share more than 1 electron. These guys share 2 electrons, so we have the double bond, right? And if we have 2 of the same atoms sharing electrons with each other, that means that this is going to be a non-polar covalent bond because they're going to share equally. But oxygen should always trigger a thought in your mind that oxygen does not usually share electrons equally unless it's sharing with its own type. But if it shares it with water, then things get a little more complicated because water has hydrogen atoms, which have very little electronegativity, and oxygen atoms, which have very high. So you don't need to memorize electronegativities for the periodic table, but the one that we do want to remember is oxygen. This guy is always trying to have more than his share of the electrons. And so the result of that is for a water molecule, the electrons shared in those covalent bonds are not shared equally, which means if we're talking about electrons, which are negative, and oxygen is pulling more of that electron characteristic towards itself than the hydrogen, that means hydrogen ends up just a little bit positive, and oxygen ends up just a little more negative. And so that's designated by the delta, lowercase, the Greek D, and we put delta plus for hydrogen because it's partially positive, delta minus for oxygen. Overall, this is a neutral molecule. But because of that, because of these polar covalent bonds, now we can have hydrogen bonds. So without polar covalent bonds, hydrogen bonds don't form. But because oxygen and hydrogen are such a big part of our molecular makeup and our just biology in general, we have hydrogen bonds happening all the time. So the hydrogen bonds happen when a partial positive and a partial negative share this attraction. They're not sharing electrons, but they're attracted because of their partial charges. Okay, this is always written as a dotted line. And the advantage of a hydrogen bond is that it can influence the structure or the behavior of something, but it's easy to break. And being easy to break is not normally considered a positive trait, but it's reversible. So that makes it nice in nature because you can do things like form DNA double helices, and then you can do things like take DNA helices apart, right? What you need to do when you're replicating DNA or when you're expressing genes. So it's nice to have a bond that's permanent like a covalent bond, and it's nice to have bonds that are transient like hydrogen bonds. Both of them have a purpose in the cell. Okay, and then we have our ionic bonds, which happen also between atoms, depending on what type of atoms we're talking about. Ionic bonds happen when atoms can ionize. All right, so we don't tend to see this with every type of atom. We don't see carbon doing this. Carbon likes to share. But chlorine and sodium are very close to reaching that full outer shell already. So sharing for them is not necessary because if chlorine can just find 1 electron, then it's golden. If sodium can lose 1 electron, even though it may lose that outer energy shell, it's happy. It finally has 8 in its outer shell. So sodium tends to give up an electron, chlorines tend to gain an electron, so the ions that form naturally, if we gain an electron, will be negative. If we lose an electron, will be positive. If we lose 2 electrons, will be 2 plus. If we lose or gain 2 electrons, it'll be 2 minus, etc. So it's not always just losing one. It just is in this example. And so now the sodium ion and the chloride ion, which both have opposite charges, these are not partial charges or polar charges, these are outright charges. Okay, so the electrical attraction between these two forms the ionic bond, which then in this case makes sodium chloride. Okay, where are we at here? We're moving on pace, I think so. Moles, valence electrons, bonds, difference between polar and nonpolar. We talked about that. What is an isomer? An isomer, I think we gave some. Did we? I thought we did somewhere. Maybe not. Well, an isomer is when we have 2 different structures but the same chemical formula. Did I not have an isomer in there? I guess I didn't. Well, I'll define it. I thought I did. Maybe it's later in the presentation. But an isomer is when we have the same chemical formula but different structures. Okay, be sure you can recognize the nomenclature of a compound and a correctly written chemical reaction. So don't read more into this than it is. I'm just asking you, if I gave you a molecule, could you tell me how many carbons, hydrogens, and oxygens? So the simplest example, and the one we'll see most, if I gave you the chemical formula for glucose and I asked you how many oxygens are in this molecule. Yes, don't overthink it. That's all I'm asking here. The number indicates the number of atoms of that particular element within a molecule. That's all we're going for there. And we're not really into chemical reactions yet in this this particular exam, but the point of a chemical reaction is just we have reactants on one side and products on the other. I don't remember if or how I asked you about that on this exam, but again, don't read more into that than it is. You start with something, you end with something. We will not be balancing chemical reactions in this class. That is kind of a fun exercise, am I right? Someone must like balancing chemical reactions. I can't be the only one.
It's not that bad.
Yeah, it's pleasing. There's a right answer. It's like math. There is a right answer, as opposed to an essay on the benefits of certain historical periods. That's all opinion. I love it when there's just a right answer. Okay, be sure you can recognize, I'm not even going to answer number 13. Know the difference between solutes and solvents. Oh, no, I keep thinking of my isomers there. Solutes and solvents, did I ever do the chemical reaction? I don't have that there, but let's mention this nomenclature, reactants versus products. We did talk about what influences chemical reactions. You guys gave me some good answers to that. I'll write them out real quick again. So things that influence chemical reactions: temperature, right? Influence chemical reactions. Temperature, concentration, right? Typically concentration of products, or sorry, reactants. pH is a big one. There was another one. Product, reactants, pH. Oh, catalyst, yeah, whether or not we have something which is speeding the reaction along. Catalysts. Okay. So solids and solvents. Solids and solvents, I like to define, as I said before, by putting both of them in the same sentence. So a solute is something that we put into a solution, or that we make a solution with, and that thing that we put the solute in is the solvent. So, for example, if we put salt into water, we're making a saline solution. Salt is the solute, water is the solvent. If we want to make sugar water, sugar is the solute, water is the solvent. If we want to dissolve something that doesn't dissolve in water, like some things dissolve better in oils or alcohols, the alcohol would be the solvent, and whatever we put in would be the solute. So water is called the universal solvent, but that's a little misleading because it's not universal, it's just probably the most common solvent in nature, but certainly there are many things that are not water soluble. So solvent does not imply that it's water, I guess was the point. Know the difference between how a water molecule interacts with polar and non-polar molecules, hydrophilic, hydrophobic. What does it mean to be hydropathic or amphipathic? So in terms of interacting with things, let's see. When we talk about amphipathic, which figured into our quiz today, it means that there's a region that has polar groups, right? So either charged groups or polar covalent bonds that lead to partial charges. And then we have nonpolar regions. Generally, nonpolar regions are long chains of carbons and hydrogens like we see in fatty acids. And the way that they interact with water is if you are a polar region, you're going to try to interact with water molecules. In this diagram, the water molecules are the little red guys around the outside. If you are a nonpolar region, right, which is hydrophobic, you are scared of water and you have to escape it. And so all the hydrophobic regions go towards the center. They put their hydrophilic or polar regions on the outside, and they form these three-dimensional structures, which eventually become, I don't think we have it there, eventually become the basis for plasma membranes too, because the plasma membranes have polar heads and hydrophobic tails that try to hide from the water. Okay, and then we talked about just some of the qualities of water, but basically the important part, basically, see what I did there, is the pH calculation, which for us is not going to involve a calculator. So if we don't involve a calculator, this becomes pretty straightforward because it limits what I can ask you to multiples of 10. And you guys showed extreme giftedness in the calculation of pH when we did it, so let's review it for a second. I know in your heart of hearts, most people really do love a logarithm. Sometimes you just don't know. So the pH equation is the pH equals the negative log base 10 of the hydrogen ion concentration. So to give an example, what if our hydrogen ion concentration was 10 to the minus 5 molar? Remember, the log base 10 of something is 10 to what power gives you that thing. So if it's 10 to the negative 5, negative 5 is the exponent. So this would be bold, just positive 5 because we've got the negative in front of the log, right? So we're looking at that exponent and we're saying that if it was just the log base 10 of that thing, it'd be negative 5. But we don't want negative numbers in our pH calculation, so we put a minus sign in front of the whole thing and we end up with positive 5. So the pH of something that's 10 to the minus 11, 10 to the minus 3, 3, 10 to the minus 1, 1. And remember what that means too. So don't just say, well, I can see the number there. Remember what it means. So 10 to the minus 5 moles of hydrogen ions means that we are talking about 1 over 1, 2, 3, 4, 5 zeros of a mole of hydrogen ions. Right? That's what the minus 5 means. That puts our number in the denominator, and if it's 10 to the 5 on the bottom, that's 5 zeros, so 1 out of 100,000. Okay, so comparing something that has pH 5 to something that has pH 3, which one is more acidic, the 5 or the 3? The 3. So as the number goes down, we get more acidic. So what does that actually mean for our concentration? It means that we have 1, 2, 3. The number of hydrogen ions is going up. Now it's a little bit maybe non-intuitive because we're using these fractions, but if somebody said, would you like 1/1000th of my fortune or 1/100,000th of my fortune? Obviously this is the bigger number, right? So as we get more acidic, we increase hydrogen ion concentration. Hydrogen ions create acidity. So if we go to neutral and then beyond down to alkaline, our hydrogen ion concentration is going down. Okay, and then the last thing was about strong acids and strong bases, and then buffers, which are weak acids and weak bases. And the difference between them is that if you have a strong acid or a strong base, you are dissociating completely in solution. So for example, hydrogen chloride is the quintessential strong acid. When you put hydrogen chloride into water, all of the chloride and all of the hydrogen ions separate from each other. 100% separation. And that means for every molecule of HCl you add, you're adding 1 hydrogen ion to your concentration, making it more acidic. The opposite is true of sodium hydroxide. That dissociates completely in solution, but each time you add a sodium hydroxide molecule, you're adding 1 molecule of hydroxide. And each time you add hydroxide, that's going to take a hydrogen ion out of solution to form water. And so that is going to make something more basic, right? If we take hydrogen ions away, then we get more basic. If we add them, like up here, we get more acidic. Okay, and then a buffer system is one where it doesn't dissociate completely. Instead, it fluctuates in this equilibrium between releasing hydrogen ions and taking them back up. So by being able to go back and forth, depending on the hydrogen ion concentration, this actually causes solutions to stay at a relatively stable pH. So this is what we see in our biological systems, maintaining, for example, the pH of our blood so that we don't have issues. Okay. Good for chapter 2? Feeling 22? Some of you are close. I'm not. There was a day, some days I feel 22, to have 2 different spaces. The matrix, which is in the very center, and the intermembrane space, which is between the 2 membranes. Okay, and so that's going to be really important when we talk about aerobic respiration. So 2 membranes, and then the second main thing that we want to say about mitochondria, we've already said that it is the so-called powerhouse of the cell. That's where ATP is generated. But an unusual thing about mitochondria is that it contains, I don't think we said this on Tuesday, it contains its own DNA. So mitochondria have their own chromosome. And it's not a nuclear chromosome like we have in our nucleus. It's a circular chromosome. It looks a lot like a bacterial chromosome. And it's thought that bacteria are that mitochondria originated from bacteria that were intracellular and then just stayed. They never left. So that's interesting. So that has some consequences for inheritance too, because you only get your mitochondrial DNA from your mom. Your dad cannot pass his mitochondrial DNA on. So of the 37 or so genes on your mitochondria, if you have a mutation or an unusual trait on one of those genes, it will be inherited in a really distinct way, because a mom will give it to all of her children, but only her daughters will pass it on to the next generation. Their sons, it's a dead end. Their sons can receive it, they can't pass it on. And some of the notable traits, or one of the notable traits that's associated with mitochondrial DNA is hearing impairment. And so that is a maternally inherited trait in most cases because of that. Okay, now let's switch over to chloroplasts, and we're talking about these at the same time because they share some common characteristics. One, they also have an inner and an outer membrane. They also have their own DNA. Say it this way. How did I say it over there? Contains its own DNA. I should have said it this way. Contains their own chromosome. It's the same thing, but just keep in mind it's just one chromosome in both cases. And then the chloroplast also, of course, this is what gives color to plants, because the pigment that absorbs light, which then transfers that energy into something usable, is found in the chloroplast. And so the chloroplast contains the light-absorbing pigments and I think maybe we'll leave it at that. We'll talk about thylakoids and granums in a couple weeks when we talk about photosynthesis. Might be getting ahead of ourselves to say it yet. Okay, and then the last thing that I wanted to look at, I'm going to skip over some of these because I decided that I want to include them in this section. But the last thing is transporting things in and out of the cell. Actually, no, it does have it there, doesn't it? Transporting things within the cell. I guess that's going to be in the membrane chapter, what I'm thinking of. So this is a different topic than the chloroplast, but transporting cargo within the cell. And the way that the cell does this, and this is going to be pervasive in our semester because we're going to see this in many different occasions at different levels. But the way that it does it is to enclose something in a membrane-bound vesicle. And a vesicle is kind of a spherical structure. It's kind of like a little miniature cell that contains something. Okay, and the way that a vesicle forms, because the phospholipids themselves do not have any structural capacity, meaning like they themselves are not going to form any three-dimensional shapes. They're just going to take on the shape of whatever's inside them. That's why the cytoskeleton is so important, because it gives structure to the phospholipid membrane. So because of that, you have to shape this to make it form a vesicle, and the way that you shape it are these things called coat proteins. Okay, and so transporting cargo within the cell, say this coat proteins form, let me try better penmanship. Coat proteins form vesicles that confuse, not confuse, that can fuse, came out weird, with other membranes. So this is a beautiful thing about using the same type of phospholipids for all of the organelles and the cellular membrane and the vesicle membranes, because one thing can bud off of this and fuse with another organelle or with the outer membrane if it's being transported outside of the cell. And so, so when we bring things into the cell, we can form vesicles. When we send things out of the cell, we can form vesicles. When we send things from one organelle to the other, in this case the ER to the Golgi, we can use vesicles. So it's a sort of central theme that we're going to see throughout the semester. Okay, so that's all I want to say about cell anatomy for now. We obviously will be talking in more detail about cellular components throughout the semester, but we're going to do that when we come to them. We don't need to get the cart ahead of the horse. Is that what you said?
So you said everything on
Carbon, classic guy, always looking for 4 bonds. Okay, so the most atoms carbon can bond to would be 4. Doesn't have to bond with 4 different atoms, because carbon is also quite fond of making double bonds, like it does in that carboxyl group there. But they are covalent bonds, right? They can be polar covalent bonds, especially if an oxygen is involved, or maybe a nitrogen. But if hydrogens are involved, they're going to be nonpolar. So this seems like a small fact on the slide, but this is really important to the basis of this entire chapter, which is that molecules interact with water differently. Fatty acids have lots of these nonpolar bonds. Amino acids have these carboxylic groups. Sugars have these types of carbon-oxygen bonds. And so understanding this slide will help you understand the activity of so many of the things that happen in this chapter. So don't underestimate it. Be able to recognize the following functional groups. So I told you that this was going to be an important thing to memorize. It would have been an important thing to have on a quiz, but that was 4 snow days ago. We just can't go back. So I would have quizzed you on this, but instead I'll throw it on the test. Amino, carboxyl, hydroxyl, methyl phosphate. So an amino group and a carboxyl group, these two guys are the basis for amino acids, right? So amino, always look for the nitrogen. Carboxyl, look for that double bond oxygen with the little hydroxyl off to the side. Hydroxyl, again, just an OH group. That's going to happen all over the place this semester. Methyl groups also plays a huge role in regulating biological systems, interestingly enough. And then phosphates, a beautiful symmetry there. The phosphorus surrounded by 4 oxygens, one of them with a double bond. All right, so just know those. There's no other way around it. Make a flashcard, make a Quizlet, whatever, however you learn best. Be sure you can know a good way to learn it, since some of you ask, how should I study or whatever. Draw these things out. Anyone can look at something and say, yep, that's it, right? We could just grab someone out of the hallway and pull them in here and say, is this a hydroxyl? Say, yeah. But if we said, draw a hydroxyl, if they knew it, they could. And actually at Lapham, there's a good chance that they would. But if they didn't know it, they wouldn't be able to do that. So being able to draw something is a good test of yourself to see if you actually know what you're doing. Know the difference between dehydration and hydrolysis. I won't call it condensation reactions, but some people do. I'm not sure why I put it on there actually. But dehydration and hydrolysis are reactions that have opposite outcomes. So if we remove water from something, we are building a polymer, right? Each time we add 2 of these subunits, we're taking a hydrogen from one, a hydroxyl from another, and that's going to form water. Meanwhile, the thing that we took the hydroxyl and the hydrogen from have connected to each other. Okay, so dehydration refers to the removal of water's contents, and polymer formation means that we are adding monomers to a growing chain of subunits. And then to go the opposite direction, we have to add water back. So lysis means to break. Hydro, of course, means water. So hydrolysis means that in the presence of water, we can break down polymers. This usually involves an enzyme doing this. If all of our stuff just broke down anytime it was in water, our cells would just be, I don't know what our cells would be, just full of monomers. There wouldn't be any polymers if they broke down just because water was present. But usually there's an enzyme that catalyzes this event. And usually an enzyme catalyzes the synthesis too. But in the process, we add and subtract water. Just FYI. Okay, carbohydrates. Okay, let's do it. Okay, we're good. Pace yourself, Peyton. One of the best advice I think I ever got in high school was from my track coach. I prided myself on having a strong finish when I ran. Really wanted to just kill it at the end. And I thought my coach would be so proud of me. And I said, Coach, I really had a strong kick. And he's like, if you had that much energy left over, what were you doing the rest of the race? It's like, oh, it really took the wind out of my sails. It was such an important lesson. Don't save it for the end, go full bore the whole way. So when I said pace myself, I thought about old Coach Rowe's advice. Why'd you have so much energy left? Okay, what are the most likely atoms to make up a carbohydrate? Well, bless you. Let's look at our favorite one. Well, my favorite one. I won't tell you what's your favorite carbohydrate. You might be a fructose kind of person. But CHO, right? And this is based on this ratio of carbons to waters. And so carbohydrates, CH and O. What are the bonds that connect carbohydrates to each other in a polymer? They are covalent bonds. They actually have a specific name. I don't remember if we really emphasized that. Maybe we did. guess I have a big green box around it. But these bonds between carbohydrates are called glycosidic bonds. This is just a reference to the fact that it's between two sugars. So glyco, like in glycogen, we see that prefix there. But glycosidic bonds, it's a covalent bond between neighboring sugars. Okay, know the places where these examples occur and are found, and what their function is in nature. So glucose, fructose, lactose, starch, glycogen, cellulose, chitin. Okay, so glucose, everywhere, right? Glucose is the monomer of so many of these polymers. So glucose and fructose are monosaccharide subunits that they're pervasive in nature. Fructose, the main fruit sugar, but glucose is the monomer that makes up so many of the things that plants and animals use, and insects, which are animals. But so glucose and fructose everywhere. But lactose, specifically, we said earlier, you all said that was the milk sugar. If we look at this chart here, starch, remember, is the main sugar that we see as a storage molecule for plants. Right? Starch is a plant storage molecule. It is a polymer of glucose. Glycogen, a polymer of glucose, that was on our quiz. And that is what animals use for short-term energy storage. Cellulose and chitin. Cellulose and chitin are similar in that they're both not digestible by most organisms. So there are organisms out there that can digest chitin, but it's not common. Most things get what they want from insects without actually breaking down the exoskeleton. It just goes in and it comes out, and they take what they want in between. But cellulose is the structural component of plants. Again, many vertebrates cannot take advantage of this unless they have microbes like bacteria in their guts that help them digest it. And then chitin, the exoskeleton of crustaceans, insects, and some fungi. Okay, what are the characteristics that can be used to distinguish between fats, phospholipids, and steroids? So fats, the example that we gave was a triglyceride. And we talked about how triglycerides are made up of fatty acids attached to glycerols. Glycerol is a 3-carbon alcohol. Fatty acids are long chains of carbons and hydrogens that are attached to the glycerol because they have this carboxylic group here that, when we release water, forms a covalent bond. Okay? And we said fatty acids can come in 3 different varieties. Saturated and unsaturated are 2 that we find naturally. Saturated is typically in animal fats, and it's long and straight. Even though it's a little zigzaggy, it's pretty straight, relatively speaking. And so multiple saturated fatty acids can stack if they cool down. And when they stack, they become solid, they become dense. But unsaturated fatty acids, like we find in plant oils, have double bonds. And the double bonds mean that we could add more hydrogens and make those into single bonds, which means it's not yet saturated with hydrogens. But because it's unsaturated, it has these double bonds that form kinks in the chain. Right? You can't rotate around a double bond. And so because of that, plant oils tend not to solidify at room temperature because they don't stack, they don't get dense. And then we have the last of the 3, which is not a naturally occurring thing, which is when we take the cis form of a plant oil and we put hydrogens on opposite sides, right? So here hydrogens are the same side, here hydrogens are on the same side, here they're on opposite sides of the double bond. And when we make a trans fat, everything is bad after that, right? The trans fats cause your HDLs to go down, which is bad because we want HDLs. They cause your LDLs to go up, which is bad because we don't want LDLs. Your overall effect on your cholesterol and your health in general is negative. So trans fat, great idea. Make a plant oil feel like an animal fat. Turned out to be bad. Some good ideas turn out to be bad. Turn out to be unhealthy. Okay, so the original question was, how can we distinguish between fats, phospholipids, and steroids? Phospholipids are different from fats because they have a polar head. They've got all these polar and charged molecules up here, right? And that makes them amphipathic. So they have a section that's interacting, or sorry, a section that doesn't want to interact with water, that's hydrophobic, one that does want to interact with water, that's hydrophilic. And so playing both sides there, we call that an amphipathic molecule. And phospholipids make up the basis for membranes. So what does it mean to be amphipathic? So that's what it means. You have both polar and nonpolar, and you can form these structures. The difference in the steroids then, steroids are based around the precursor cholesterol. Would have been nice to have that answer on your test, on your quiz. Cholesterol is the precursor for the two main sex steroids, estrogen and testosterone, which differ by a very small number of chemical changes, but have dramatically different outcomes. Okay, then it did have a couple other questions that aren't in that PowerPoint. What are the health facts associated with HDLs, LDLs, and TGAs? And we can summarize that pretty quickly. You don't need to memorize the numbers associated with those because those numbers change, and it depends who you ask, and it depends on your age. So it's a complicated picture, but what we do know is HDLs, good. LDLs, bad. Triglycerides, bad. And so we would like to have high HDLs, which is primarily the result of activity and exercise. We would like to have low LDLs and TGAs, which are primarily the result of diet and lifestyle and partially affected by your genetics. So some people have less control over their LDLs and total cholesterol than others due to just family history, basically. Some things we can't change, sadly. All right, proteins, getting there. Oh, almost there. Boy, I didn't pace myself. Now I'm not even gonna finish strong. My coach would be ashamed of me. Let me see if there are any that would not be obvious here. Structural features that all have in common, that bonds that connect them, peptide bonds, levels of protein structure. We mentioned that. Primary, secondary, quaternary, tertiary. Don't worry about how mutations are named. I'm not going to ask you what number 13 says. That is something I used to emphasize, and I didn't realize I left it on my review here. And then nucleic acids, a short section here. So the nucleotides in DNA and RNA. DNA has, surely I gave you this list. When we did that, DNA is A, C, T, and G. RNA is A, C, U, and G. So the difference being T and U there. The bonds that occur in the molecule in the double-stranded polymer. So we have phosphodiester bonds connecting each nucleotide within a strand. We have hydrogen bonds connecting nucleotides on neighboring strands. And how are the sequences of the two strands related to each other? Whenever we have a guanine on one strand, we have a cytosine on the other. That's misleading the way they label that. Whenever we have an adenine on one strand, we have a thymine on the other. So you know this to be called base pairing, which we're not going to get into too much later. But the relationship is that an A is always attached to a T, and a G is always attached to a C. How are we feeling? Okay, that was a tornado of a review, but hopefully we touched on all the highlights. And if you have questions as you go through your